Okay, so now that we've looked at what inheritance is and how it occurs, we're going to look at Punnett squares and how we can predict what the offspring of a particular breeding pair might be. Okay, so for these, each one we look at a specific trait at a time. So in this Punnett square that we have here, we're going to look at yellow pods versus green pods. And we can see that in this diagram, we have been given that the parent cells are homozygous dominant because we can see that the green pods are a capital G. So this means that this is the one that will be seen if it's in combination with a recessive, and then homozygous recessive with little g, little g. So what we do is we draw up our actual Punnett square. So we draw up the square, we put in our possible combinations. Okay, so we draw up the grid, and we can see that we put both options here. So across the top we're going to put P1, which was homozygous dominant, and then down the side we have put P2, which was homozygous recessive, and we put each of the gametes, because this would represent the possible gametes from these individuals, along here to set up our table. And then we fill the table in with the possible combinations, and we always put the dominant letter first. So in this case, if we're combining the little g with the big G, it would form the combination of big G, little g. And then the next one would be the same, because we have a big G and a little g. And then if we take the little g here and the big G there, we get that. And then we get that. So you can see that in this case, this pairing would produce 100%. All of the offspring would contain big G, little g. So they would all look green. Their phenotype would be green. Okay, because the dominant trait would be the one that's seen, and that's the green pod. But their genotype is going to be big G, little g. So the phenotype is green, but their genotype is different than the parents. It's big G, little g. Okay, so they would all be heterozygous green pods. If we then looked at an F2 generation, so we take two of the F1 generations and combine them together, then that means across the top we would have our big G, little g, and then down the side our big G, little g. And when we combine these, we will get a different set of allele combinations. Okay, so in this case, when we combine big G, we get a homozygous dominant. Big G, little g, we get a heterozygous dominant, remembering that we would usually write this with the big G in front. Then we get big G, little g again. These ones are the same. And then this last combination would actually end up being a yellow pod because both recessive alleles combine, giving us the only homozygous recessive. So this is the only one that would show the recessive trait. So our phenotype would be 75% or 3/4 green for the phenotype, and 1/4 or 25% yellow. Okay, but the genotype combinations would be different. We would have 25% that would be homozygous dominant, 50% would be heterozygous dominant, and then 25% would be homozygous recessive. So we can do this with any number of combinations, and we will look at different ones as we practice these. But so here is another example. So this one on the left has no pictures. So we put the gametes across the top. Okay, so the gametes across the top for one parent, the gametes for the other parent down the side, and then in the middle we fill in the traits. So this one would be big R, little r. Okay, so we don't know what this might is. This is red flowered peas. Okay, so this is looking at red flowers, and the big R stands for a dominant trait of a red flower, and the little r stands for a white flower. So when we combine these, we can see that in the first generation, the F1 generation, all the flowers would be red, but they would have a heterozygous genotype. Red phenotype, heterozygous genotype. If we take a generation from these and we take 2 of these and we breed them together, we can see that we would end up with 3 red flowers out of 4 as a possibility, so 75% of them would be red, and then 25% would show a white flower. And that would be our phenotype, but our genotypes, we would have 50% heterozygous dominant, 25% homozygous dominant, and 25% homozygous recessive. So I want you to have a go in your notebooks and look at this example here. So we can see here that we are combining a black chicken with a blue chicken. Okay, so black is homozygous recessive. The blue is heterozygous dominant. And we can see here that we have produced 2 black chickens, 2 blue chickens, and 2 black chickens. So I want you to specify the genotype and the ratios for each of these, and then come back and check your answer. Okay, so hopefully you found that you had big B, little b for the first blue chicken, and then big B, little b for the second blue chicken, and then our black chickens will obviously be little b, and little b, little b. So if you have that skill down, great. We're going to be doing a lot of practice in class. So I'll see you again soon.